High blood pressure or hypertension is a very common condition that can arise from physical activity, stress, or certain disorders. This, in turn, leads to arteriosclerosis and increases the risk of cerebrovascular disease. Why and how hypertension triggers vascular remodeling is not entirely clear. Scientists have shown that macrophages, a type of white blood cells that kill foreign bodies, are involved in the transformation. Specifically, the macrophages accumulate within blood vessel walls from outside the vessels and cause chronic inflammation. Against this backdrop, researchers from Japan and Canada, in a new study, recently investigated a mechanism known as excitation transcription (ET) coupling in vascular smooth muscle cells. By unveiling the mysteries behind the ET coupling in these cells through experiments spanning single cells to whole organisms, they successfully linked the ET coupling mechanism with vascular remodeling. The study, published in the Proceedings of the National Academy of Sciences (PNAS), was led by Junior Associate Professor Yoshiaki Suzuki, Hisao Yamamura and Yuji Amaizumi from Nagoya City University, Japan, and Gerald W. Zamponi and Wayne R. Giles from University of Calgary, Canada. Various types of cells are known to undergo ET coupling. In neurons, for example, an excitation in the form of calcium ions, Ca2+, entering the cell through calcium channels activates certain transcription factors and enzymes. These, in turn, trigger the transcription of various genes. Meanwhile, although ET coupling also occurs in vascular smooth muscle cells after an influx of Ca2+ under high pressure, not much was known about how it happens, what genes are triggered, and the role it plays in our bodies. Through detailed experiments in individual cells, cell cultures, and live mice, the team found that a specific protein complex found in caveola is a key player in ET coupling in vascular smooth muscle cells. They proved that this complex, referred to as CAV1, 2, CAMKK2, CAMK1A, is formed within caveola, and both CAMKK2 and CAMK1A are directly activated by Ca2+ entering through CAV1. 2 when subjected to certain stimuli, such as high pressure. Moreover, they showed that this complex activates a signaling pathway that phosphorylates a transcription factor called CREB, which ultimately leads to an increased transcription of multiple genes. By taking a detailed look at the genes promoted by ET coupling and observing their effects when blocked or amplified, the researchers made some important discoveries. Firstly, some of these genes were related to chemotaxis, the phenomenon by which cells' movement is triggered and directed by chemical stimuli. This helped explain the accumulation of macrophages in blood vessel walls from outside the vessels. Additionally, these genes promoted the remodeling of the medial layer of arteries, where vascular smooth muscle cells reside and control blood flow through contraction and expansion. Taken together, our results explain how ET coupling caused by high pressure in vascular smooth muscle cells can modulate macrophage migration and subsequent inflammation, altering the vascular structure, explains Dr. Suzuki. The findings of this study have important implications regarding anti-hypertension drugs. For one, they explain why medications like nicardipine, a classic calcium channel blocker, prevents vascular remodeling and the progression of arteriosclerosis. This not only fills an important knowledge gap in medicine but also presents several potential drug targets for treating or preventing vascular remodeling, such as the constituents of the CAV1, 2, CAMKK2, CAMK1A complex. About 40 million people suffer from hypertension in Japan alone, and are at high risk of stroke, end-stage renal failure, and vascular dementia, says Dr. Suzuki. Understanding the mechanisms behind arteriosclerosis is, therefore, very important for reducing the incidence, progression, and recur. A molecular complex of CAV1, 2, CAMKK2, CAMK1A in caveola is responsible for vascular remodeling via excitation-transcription coupling. Proceedings of the National Academy of Sciences, 2022. 119, 16, doi. 10. 1073, PNAS. 2117435119 Nagoya City University. https://ift. tt. vp70ydp. Accessed April 22, 2022. Thanks for watching. Please subscribe my channel for more videos.